Good morning, everyone, or good afternoon. This is ASM's Microbial Minutes, Too Many Genomes Edition. I'm Julie Wolf, the Science Communication Specialist here at ASM, and we are going to look at recent literature to answer some questions about microbiology-based problems. Those questions are asked on slide 2. So we are going to look at some research that suggests ways to improve application of an increasing number of microbial genomes. And then we're going to discuss a story that looks at where HIV-infected cells hide. So let's dive in and look at that very first question, how to improve applications of an increasing number of microbial genomes. This is on slide 3. So here we're going to look at 2 stories from Nature Biotechnology. The first one is titled 1520 Reference Genomes from Cultivated Human Gut Bacteria Enable Functional Microbiome Analyses. And so what this particular group did was to take a number of different fecal microbiota samples from people throughout the world of different ethnicities and different geographical locations and do whole genome sequencing in order to get more reference genomes to be representative of the fecal microbiota that are inhabiting our gut microbiomes. And this is important because although there are a lot of microbiome studies being done, much of the characterization of who is there is done by looking at the 16S ribosomal RNA gene sequence to be able to tell which lineages there are, but it doesn't give the entire sequence information of all the different microbial characteristics. And in fact, when you look at the reference genome for what is within the NCBI database, only fewer than 4% of the genomes that are deposited there belong to human gut microbiota. So what this group did was to use 11 different culturing conditions, and they isolated over 6,000 new isolates. Some of those were redundant, of course, and so when they took out redundancies, they saw that this collection represented 120 non-redundant draft bacterial genomes. This contributes to that database and to databases where these sequences are deposited, at least 264 new reference genomes, which, as you can see in the picture on the right, are representative of many of the different phyla that are normally associated with the gut microbiota, but now instead of having merely partial sequences, we have entire gene sequences to be able to look for different characteristics. However, this doesn't answer the question of how to improve application of all of these different genomes. Looking through all of these different genomes in combination with this study and all the studies that are out there in these different repositories can actually be quite time-consuming for computers to look for a particular gene sequence. And so the other story that was also published in the same issue of Nature Biotechnology really addresses this question. So we'll look at that on slide 4. Here, we're going to look at a study with the take-home message where a new search method increases the search speed in genome databases. And being able to search for something is really something that a lot of us take for granted. When you go to Google and look up a particular question or a topic, it pops up very quickly. And that is in part because it uses an indexing method, which the research scientists in this study applied to the microbial genomes. So they restructured the way that the data is stored, and they called this a bit-sliced genomic signature index, or BIG-C, or BIG-SCI, where they indexed nearly 450,000 microbial whole genome sequence data sets. And the way that they indexed it ended up requiring over 4 orders of magnitude less storage space, which allows searches to occur much more quickly. So if you look at the figure on the right, this is an explanation or a schematic and an example of how this sequencing is stored and searched for. So we have 5 sequences, our FASTQ formatted sequences that have a particular ATGC nucleotide sequence. And these are broken up into various length short sequences within. In this case, we have 3 MERS. So we have the FASTQ1 is ATC, TCG. And you can see that those different sequences are found in various members of the sequence family that we've deposited. So those groupings of nucleotides are then assigned a number. So in this case, we are indexing these various 3 MERS with 0 through 9, and each of those numbers represents one of those trimer of nucleotides. And you see that they repeat. So for example, in FASTQ1, we have ATC, and also in FASTQ5, the second of the sequences is ATC. And so in both of those, we see that there is a 9, and I think that 9 here is what represents the ATC sequence. So now, when someone goes to look for a sequence, and we're looking at step 2B, the query is for an AAT. And so AAT is represented by one of these numbers, and it's much faster to look through these numbers and then find the associated FASTQ sequences, rather than the previous style of searching, which is something you might be familiar with if you've ever used BLAST, which is basic local alignment search tool available on PubMed. And in this case, it queries the entire the entire sequence of that particular FASTQ sequence in order to find which of the sequences within that database have that particular sequence. And so by using this index method, it allows the researchers to much more quickly look for sequences that are there present in a database. And this was really found to be useful around the time that the MCR1 gene was being found in human isolates. MCR1, you may know from previous sessions, encodes for resistance to the antibiotic colistin. And this was found in a human isolate around 2015. And folks working with Zaman Iqbal, the senior scientist here, they were looking for whether any isolates within the database at Public Health England, which is where they work, might have this gene already. And so they wanted to query the entire database to see maybe somebody who had already had one of these isolates but hadn't needed colistin, or this hadn't happened to have been found, already had this MCR1 gene within its sequence. And it took 256 computers an entire weekend's worth of search time in order to identify whether those sequences contained the MCR1 sequence. And now with this new index method, you would be able to search through those types of databases much faster with, again, like the quickness of doing a Google search, which is very useful. And so on the next slide 5, we'll see that Iqbal himself shared a lot of these results. He summarized some of the uses, comparing plasmid relatedness and seeing that there are sequences from certain plasmids that, of course, are going to be shared between the very promiscuous E. coli, Salmonella, Shigella species, which tend to share plasmids, as you can see in the tweet that he shared. But some of those sequences can also be found in very disparate types of species, and that could be indicative of a number of different types of horizontal gene transfer events that have occurred. This was written up very nicely in The Atlantic by Sarah Zhang, where she highlighted some quotes from Iqbal as well as from researchers who have been using this database. So we'll provide links to all of the papers that we've discussed here, as well as the database itself. There's a nice link for a demonstration if you want to kind of try it out for yourself and do some searching. And it's been available because this was published as a bioRxiv paper and has been used. This Big Sci has been used by a number of different researchers already in this piece in The Atlantic. Tara Smith, who is an epidemiologist at Kent State University, says that Big Sci is a fantastic idea, although the tool is only as good as the data that go in. The genomes we choose to sequence are very biased, she says, often towards serious clinical infections from patients in research-intensive hospitals in big urban centers. And I think this really ties in the first study, showing that we can very quickly add to these databases by doing non-selective or very broad searches for what types of microorganisms are around. And as those are added, hopefully the ability to search through these very large data sets will be made easier by tools such as Big Sci. So our next question was, where do HIV-infected cells hide? And so we're going to go to slide 6 to look at this one. Here we're going to look at a Nature paper. The take-home message from this paper is that a new assay, distinguishes intact versus defective HIV provirus infected cells. So it's important to understand the life cycle of HIV. HIV infects an individual and then attacks the CD4 T cells. And in many of those T cells, it will become replicative, making progeny virions, which will eventually break open and kill that T cell. but in a small portion of cells, it will not begin to make its progeny virus right away. And as part of the life cycle, the RNA virus, the genome, the RNA viral genome of HIV is converted into DNA, which then incorporates randomly into the host genome. And you can see an illustration of this on the right-hand side. So in those T cells that have latent infection, that are not actively making new virus, it's a little more difficult to attack and detect those for a number of reasons. Most of the antiretrovirals that we have currently that we treat HIV-infected patients with, those attack the cells that are actively propagating the virus and making more virus. And so these quiescent or latent proviruses, they escape elimination by the use of those antiretrovirals. Understanding where those reservoir cells, the cells that could potentially begin to reactivate again, is necessary for any assessment of an HIV cure, because the ability to detect which proviruses could potentially restart the infection again, begin to make progeny viruses, and then infect the remainder of the CD4 T cells that haven't been infected, and begin the cycle of viral replication again, is important to understand whether or not any treatment of elimination of the virus within the body is going to be effective. Now, finding that reservoir is really hard because, first, many of those proviruses that incorporate are actually not very good. They're defective. They are not able to make new HIV viruses. And if we're using molecular means, such as PCR, we're not going to be able to determine if there is a single nucleotide difference that might inactivate the transcriptional activator, for example. And in that case, molecular means would not be able to differentiate. One assay that some people use to detect these latent viruses is to take out different white blood cell populations from patients to eliminate any actively reproducing virally infected cells, and then to stimulate with various cytokines or stimuli that might reactivate any proviruses that could potentially reactivate and begin propagating. The problem is that those stimulations, they're only given for a certain duration, and it's possible that you might not estimate the actual number. You might underestimate the number of those viral reservoir cells. And so this new method, as described in this paper, separately quantifies the proviruses into intact and defective proviruses. So how do they do that? Let's look at slide 7 to find out. Here we can see that they took 28 patients, people who were infected with HIV, and these patients were part of a clinical study, and they donated their white blood cells, from which the researchers analyzed 431 different provirus sequences. So different sequences that had incorporated into the T cell, the CD4 T cell genome. Keeping in line with what has previously been seen in the literature, they saw that the vast majority of these were totally defective, unable to propagate and make new HIV variants. So that would be 97.6, had either a deletion or a hypermutation, which made that viral provirus defective in some sense. And only 2.4% of those proviruses were actually intact and could potentially reactivate. So what they used was a technique called digital droplet PCR in order to separate a variety of different sequences. So digital droplet PCR is really good if you have multiple different sequences and you want to detect a minority sequence or a minor sequence. The sequences into smaller subgroups, which increases the probability of detection of any minor sequences that might not be detected because of competition for the PCR reagents, for example, or preferential amplification, what have you. And in this case, they used the characterization of the proviruses. As you can see in the pie chart on the right-hand side, they saw that the proviruses were able to be grouped into various settings. They had deletions either on the 5' end or the 3' end, or certain hypermutations that were particularly associated with defective viruses. And so what the the assay involves is a dual allele PCR. So they do two different PCRs, one that would detect differences in some of the sizes, and one that is going to be able to amplify regions that are particularly susceptible to that hypermutation. And then they can look at the differences in length, product length, for any potential deletions. And they have these allelic discrimination probes. So these are fluorescent probes that will become fluorescent when they find their cognate sequence. So if there is a particular G to A mutation that is associated with a defective virus, when that probe finds its cognate sequence, it will then light up, and that can be detected by various means. In this case, you can see in segment B below that there are different fluorophores that measure either the deletions on the 5' or the 3' side or the hypermutation. And from using these various probes, they are then able to separate different quadrants of either hypermutated, intact, or deletion proviruses. And from that, they're able to back calculate what the ratio or proportion of these different sequences in the population are. So they applied this, as we'll see in the next slide, slide 8. And here they looked at various memory T cells. Memory T cells are cells that are often inert. They have been activated and then undergo a maturation phase so that they still exist and can reactivate in the event that they see a cognate antigen and that they need to attack, right? So if you get the flu once, it will attack that same influenza strain because your body recognizes it. And so they looked at the central memory, transition memory, and effector memory, which are different, that have different functions within the immune system. And they were particularly looking for those red dots, right? Those are the intact proviruses that can potentially reactivate and are the ones that are essential to eliminate for an HIV cure to be effective. And you can see that there's a larger proportion of these in the central memory cells. What this assay really does is not say where entirely those HIV-infected cells live or hide, but it does give us a tool by which researchers can now go forward and continue doing more clinical population studies to look at people with HIV to better determine where and what type of cells are hiding those HIV latent proviruses. And so this was written up into a press release from the funder, HHMI, Howard Hughes Medical Institute, although the research was done at Johns Hopkins. And here they have a lovely quote from the senior scientist, Siciliano, who is actually the scientist who first realized that there is this latent reservoir within patients. And now he and his group have developed an assay slightly over 20 years later to better differentiate the types of proviruses that are in these T cells. And what Siciliano says is we may still be a long way from a cure, but now we can at least measure our progress. And so going to slide 9, we can summarize what we've talked about in this Microbial Minutes session. We discussed a new search method that will increase the search speed in genome databases, very useful tool for people working in large data sets. And we also discussed a new assay that distinguishes intact versus defective HIV provirus infected cells. I'd like to thank you for listening. As always, if there's a study that you want to hear us discuss on Microbial Minutes, leave us a link down in the comments section. And I will be with you next time. I'd like to thank Ray Ortega for helping to coordinate and produce the show. I'm Julie Wolf, and I'll speak with you next time on Microbial Minutes.